Michael.
Shenanigans with the computer, yes.
You don't usually mention eco-lunch, but I'd like to just mention that this
Please.
coming Monday, I'll be talking about can ecologists lead humanity into a sustainable future? So people might like to come.
And that's Monday at what time?
12.
At 1.
12.
Oh, at 12. Great, okay. Can they?
No question.
Sounds good. Okay, any other announcements? All right, it's a special pleasure for me today to introduce Bret Payseur, who comes to us from the University of Wisconsin at Madison. He did his undergraduate degree at the University of Colorado. He then did a master's at Northwestern University, where he worked with Ann Yoder, our seminar speaker for next week. And then he joined my lab as a graduate student at the University of Arizona. And while he was a fifth-year graduate student, and this is something you all of the graduate students here can aspire to, he applied for a couple of faculty positions, and he got the ones that he applied for, and he accepted his current position at the University of Wisconsin in the Department of Genetics. But he already had a postdoc lined up to work with Andy Clark at Cornell, so he then went there and did a 2-year postdoc with Andy Clark before moving to Wisconsin, where he's been ever since and is now a full professor. I still have tremendous fond memories of the time when Bret was in my lab, because as much as anybody I know, he just loves talking about science. He's got this infectious enthusiasm for it, and he's very scholarly, and like all of the best students, I've learned more from him than I think I ever taught him. He's done a lot of work on the evolution of recombination rate, on the genetic basis of reproductive isolation, and more recently on the evolution of body size, combining experiments in the laboratory with genetic methods and QTL mapping, and then mice that have come from natural populations. And that's what he'll tell us about today, something about big mice on islands. So please join me in welcoming Bret.
Well, thank you, Michael, for the invitation to present here and for that gracious introduction. And I dispute the direction of who learned more from whom, for sure. But I count myself as extremely fortunate to have been one of Michael's students. Okay, so it's an honor to be presenting to this crowd. As biologists, we're all, I think, captivated by biological diversity that can be measured in a number of ways. This beautiful slide was made by my postdoc, Amy Dapper. And often we think about this in terms of differences between species, which are really exciting to look at. But it's worth remembering that all of the evolutionary differences between species ultimately start as differences within species, which means that the comparison of phenotypic variation within and between species provides a powerful way to understand mechanisms of evolution. The strategy we follow in my lab is built on this principle. We start by documenting variation within and between closely related species. We apply concepts and methods from genetics and genomics to hopefully understand in the end mechanisms of how evolution works. We have a number of lines of research in the lab that use this strategy. The first one that I've been working on for the longest is the process by which one species becomes two. And we're integrating genome-wide studies of gene flow in hybrid zones with the genetic characterization of reproductive isolation phenotypes in the lab to try to come to some understanding of the genetic basis of speciation. As Michael mentioned, I'm also interested in the evolution of recombination as a trait that itself affects evolution. Recombination rates evolve on short time scales, and we don't understand, I would say, too much about why and how that works, and so we're really interested in that. But today I want to talk about big mice on islands, and for this I've been very fortunate to work with a talented group of students, including Melissa Gray, Michelle Parmenter, Mark Nolte, and Pei-Ching Jing, and then faculty, and Lauren Brooks as well, and then faculty collaborators, Carl Groman, Colin Dewey, and Chris Vineyard. And so all of the work that I'm going to describe today was really done by them. Biologists often refer to islands as natural laboratories, and there are many reasons for this that many of you will be familiar with. But one of the reasons is that island populations are unusual. Just taking a quick survey, we note that we see things like flightlessness, classic changes in morphology that tell us interesting stories about evolution, changes in behavior, and one of the most striking differences between island and mainland populations is in the evolution of extreme body size. So looking at species of mammals, in 1964, Foster published a classic paper in which he provided this table tallying, just taking a bunch of different species of mammals and asking whether the populations that colonized or lived on islands were different in size from their mainland relatives. And he found that in a number of cases they were. And there was an intriguing pattern that came out of this potentially that larger mammals tended to get smaller in body size when they arrived on islands. And smaller mammals, particularly rodents, tended to get bigger. And rodents actually showed the strongest signature and continue to show the strongest signature for what Lee Van Valen later called the island rule. I got excited about this island rule because it raises the prospect that we can identify common mechanisms that govern the evolution of body size by comparing island and mainland populations. And ecologists have been working on this, wrestling with this problem for decades, and have identified a number of factors that are correlated with changes in body size. But despite this proliferation of exciting ecological studies, there is actually very little data that demonstrates that the evolution of extreme body size has a heritable basis. So we got interested in attacking this question from a genetic perspective, and mice here are great for all the usual reasons that we have these amazing genetic and genomic tools. We have detailed knowledge of growth pathways. Mouse body size has been studied for 100 years, and so we understand quite a lot about that, at least in the lab. Much to the consternation of conservation biologists, house mice are some of the most successful invaders that we know of, and so that means they've colonized islands all over the world, so there's a chance for natural evolutionary replicates to look at here. When I first started thinking about this, I literally compiled body weights from the literature of house mice anytime they were caught on an island. And I also asked a bunch of mouse biologists who spend more time trapping mice than I do, which isn't that hard actually, but, to tell me where the biggest mice in the world were. And the literature research and the experts all said the same thing, that there's this crazy island in the middle of the South Atlantic located here, which some people have called one of the remotest islands in the world, that's uninhabited by people. And on this island, there are house mice that we think arrived there via sealing ships approximately 200 years ago from historical records. Someone pointed out to me that when I show this, I should really emphasize the sealing ships, because last time I gave this talk, somebody said, "Wow, that's a mouse?" It's not. But our projects on the genetic basis of size and seals haven't progressed as much. So anyway, Gough Island seemed like the place to start. And there is a terrific group of South African conservation biologists led by Peter Ryan who have been studying these mice for some time. They have a lot of unusual traits. One is that they feed on endangered birds to the point where the British government is now raising money to exterminate all of the mice on the island, which I support in principle, but also a little bit sad about, although I don't expect to get much sympathy there. And so there are interesting questions about the evolution of diet and other behaviors actually that we're starting to look at. But I wanted to start with something I thought would be a little simpler, which is body size. And so this is just a schematic showing, or a figure showing average body weights, again from the literature, for a bunch of mice from a bunch of different islands. These are all the same subspecies, Mus musculus domesticus, and a few from the mainland. And we really need to get the Nachman lab numbers on here because I suspect the Gough Island mice might not seem as unusual once we do that. Oh, they will still? Okay, good. Then definitely we should put them on there. But anyway, these are big mice, okay, and most people agree that they seem to be the biggest house mice that are on record. So that seems like a good place to start. They also live on an island that's remote. It's all good. So here's a picture of Gough Island mouse next to a mainland relative at the same age, and I probably don't have to say which is which. You can figure it out. But you know, the one thing to consider is that this is a size difference that we think evolved over a pretty short time scale. So that this could be very rapid evolution of body size. Okay. So to begin, Peter Ryan was gracious enough to actually, they've been trapping these mice for many years now. And so I emailed him and just said, I would love to study these mice. And amazingly, he just caught a bunch of mice and sent them to us in Madison, which I'm extremely grateful for. This isn't exactly the route that they took. If you've ever had trouble going through security at the airport, just imagine what happens when you say we'd like to bring in 50 giant carnivorous mice. It's not, I don't recommend it. But anyway, so we ended up getting 46 mice that started the original colony, and then we got a wild-derived mainland strain called WSB, which we just ordered from the Jackson lab. And that's descended from a population on the east coast of North America. So these are the comparisons that I'm going to talk about today. So the first question was just to see what happens when you put these mice in a common environment. Because as I mentioned, there wasn't a lot of evidence that the island weevil had a genetic basis, and it looks like it does, at least in this example. So we started by, Melissa Gray was with a big team of undergrads weighing mice for 16 weeks every week. And these are the growth curves that we got. And so you can see that Gough mice are possibly a little heavier when they're born. But they definitely, in both sexes, grow faster. This is the growth rate, especially during about the first 6 weeks. Okay, so it didn't have to be this way. There are other ways to get big, but it looked like, at least compared to the WSB strain, that there was an acceleration of growth early on, which is helpful. That already helps us hone in on what's happening. But we were interested in actually mapping this trait, and so we took a quantitative trait locus approach where we generated F1s between Gough Island mice and WSB. These were actually partially inbred when we did this cross. And then the F1s were crossed to each other, and through this magical process called recombination, you generate F2s that are genetically unique, and ask what their body sizes are, compare that to which parent they get which genomic region from to identify an association with the trait. So all of the mapping results I'm going to show you are based on this kind of an approach. And so when you do this, you first want to see that there's phenotypic variation to map. And what I'll just guide your eye towards here in terms of both body weight and growth rate is this shaded, this shading, which shows the distribution of body weights and growth rates across the F2 population. And I'll just point out that this was an incredibly laborious process to which I owe Melissa and the team a lot, because now they're weighing mice every week for 16 weeks, but the number of mice that they weighed was close to 1,300, because we wanted to get at, try to find some regions of the genome that had modest effects if they existed. And then the next step is to genotype them, and because we're working on house mice, we were lucky there are resources available. We used this SNP chip that had 78,000 SNPs, which is about 77,000 more than you need to do QTL mapping and F2 cross. So we were in good shape there. A large number of them were informative. And so the approach we took was we have the growth curve for every one of 1,200 F2s after doing some very careful data cleaning. And we can actually map the growth curve itself. So we can map, weekly weights, but we can describe the growth curve in some functional form and map that, which hopefully gives us a little bit of a better sense of what's happening. So combining information across ages and also accounting for effects, well-known effects on body size of mother and sex. Okay, so here are the results for the QTL mapping. What's being shown here is just the QTL that we found. Each of them is attached to a chromosome, which is good. And on the y-axis is a measurement of the statistical evidence that there's a QTL at that location. And all of these are highly significant QTL. And so there are 8 that we found for body weight, including possibly 2 on different pieces of chromosome 10. And we did the same thing for growth rate, which is just the derivative of those body weight curves. And there we got 11 QTL. Some of these overlap with the body weight QTL, including these ones on 10, and some of them are distinct. Okay? But this is our first picture of the genetic basis of the island rule for a sample of size 1. In terms of the effects of QTL, we were really interested in that as well. And we were a little bit surprised to see that they're very similar in males and females. There's no evidence for an interaction between sex and genotype, despite the fact that, as you probably noticed before, males and females have different growth trajectories. They're also similar among mice with different mothers, despite the fact that there's clear evidence for maternal effects in the data, just like there is generally in mice. They were modest in magnitude, so we're looking at explaining between 5 and 15% of the variance among F2s. So actually, some people call that modest, some people say those are big effects. It depends on what school of thought you come from. They're mostly additive. We don't see much evidence for dominance and not much evidence for epistasis yet, although figuring out how to do scans for epistatic QTL when you're looking at multi-dimensional phenotypic data like this is a little bit of an unsolved problem. So there could be some epistasis hiding in there, but the effects at this point that we found are mostly additive. And we also noticed that Gough Island mouse alleles are associated with increased size. And this got us thinking about this test that was proposed by Alan Orr years ago. The rationale was laid out for it by Alan Robertson, actually, before that. But the idea is pretty simple, that if you imagine body size evolving, or any trait evolving neutrally with no persistent force pushing in one direction, once you condition on the difference in the size between the parents, what you expect is a random distribution of sizes for the QTL, so that the effects of you should have just as many QTL. The Gough allele should increase size for just as many loci as it decreases size, okay? And so this is the neutral prediction, and you can do something that's basically like a sign test to look at that. And at least for the weight QTL, where this test is easiest to apply, we don't see this pattern. You see that all of them, the Goff allele is associated with increased weight in all cases. And of course, this is not a significant test, but it's the most extreme result you can get with a QTL. And we're working on how to perform this test with the other QTL I'm about to tell you about. So, this is, observation is not, I think, definitive, but at least suggestive that the difference in size was driven by selection, whether it was size itself that was targeted or some trait with which it was genetically correlated. Because we're working on house mice, we can compare this evolutionary trajectory to what we see in classic experiments where mice were artificially selected for size. And the first thing I want, so this is work that was done by many people, but the genetic characterization has been led by Jim Chevery's lab. And first observation I would make is that these are really different, just to be honest. You know, if you look at the axes here, this is just nuts, right? It's a 60-gram mouse. So artificial selection, it turns out the rumors are true, it works. You can really make things highly divergent. But I think it's interesting to compare the genetic architectures here. And what we see is that the numbers and effects of QTL, when you try to account for differences in sample size and all of that, are pretty similar. Modest in magnitude, additive, not a lot of sex differences, sex by genotype interactions. But the QTL themselves are mostly different. So, I think what all of this points to is the startling realization that body size is a complex genetic trait, which is maybe not that startling. But it is interesting to see that the properties of natural populations are to some degree recapitulated by these experiments in the lab. But they probably won't get us to the genetic changes, specific ones that matter for nature. But we can do a lot more than just look at weight. And my student, Michelle Parmenter, led the effort to look at the evolution of the skeleton. She started out by taking 16 measurements from x-rays at 3 different time points. And this is, again, Michelle was looking at over 800 F2s. And so this is an incredible amount of work. And to summarize what she found, if you compare Goff Island mice to at least to this mainland strain, what you see, not surprisingly, is an overall expansion of the skeleton. Sort of every dimension that we look at is bigger. There is interesting relative lengthening of the skull. So the Goff Island mice are more dolichocephalic. I love that word. I just wanted to. I woke up this morning and thought, I really want to say dolichocephalic. And so we don't know why, but that's an interesting morphological change potentially. And there's also a disproportionate expansion of the femur and the humerus relative to the tibia and the ulna. So these are fun examples of morphological evolution. What Michelle did first was just to look in the F2s. We know the skeleton travels as a package in general. And these are the correlation matrices between all of her traits, where blue indicates positive correlations, with dark shading being stronger. And the main point I want you to get here is that there are at least modest up to strong correlations between these traits, positive correlations. And interestingly, if you look at 5 weeks, 10 weeks, and 16 weeks, you see very similar patterns, suggesting that these these developmental correlations were laid by 5 weeks. Okay. So we can go through the same exercise and map QTL for skeletal evolution. So there's a list of phenotypes here on the y-axis and then chromosome on the x-axis. And we have 3 different time points that are denoted by the colors. And you can spend a lot of time staring at this. It's a pretty interesting depiction of the genetic architecture, but I just want to point to 2 things. We find both QTL that are restricted in their action, including this one in the head. And we also find some QTL that have very broad effects across the skeleton that presumably reflect global modifiers of the growth rate. So again, there are a lot of details here to consider, but that's the big picture. To summarize the genetics of skeletal evolution, we have multiple QTL contributing each trait, although not generally as many QTL as we see for body size. Each QTL is explaining between 9 and 23% of the difference between island and mainland strains. We have some QTL affecting particular regions and others that are global modifiers of body size. And those usually overlap with QTL for body weight, so that actually makes sense. Okay, so that concludes the QTL mapping section of my talk. We're obviously interested in going further than identifying these big genomic regions. And, but this is, I think many of you recognize, a big challenge to go from QTL down to the causative genetic variants. That's where we're headed. We're taking what has now become sort of a classic approach of building congenic strains, moving QTL from the Gough Island mice onto the background of WSB to characterize the processes and see what's changing in terms of growth. But something we can do in the meantime is look at differential expression of genes. And the reason for doing that is that when you think about these quantitative changes in size, right, and a shifting towards an acceleration of growth early on, it's one of those situations where you really would expect changes in the timing or location of expression of genes. It's sort of a classic situation to predict regulatory evolution. And so we started, Mark Nolte started to look at this, doing a relatively large RNA-seq experiment for 5 females and 5 males each of Gough Island mice and WSB. He sampled at 4 weeks the gonadal adipose depot, the hypothalamus, and then the liver at 4 weeks, but also at 2 weeks and even an embryonic time point. And these were strategically chosen based on what we know about development and what we might expect in terms of transcripts influencing body size. So remember, what we're trying to do here ultimately is understand what's happening with this shift in growth rate. We were kind of surprised, although I don't know if you will be, to find that how much differential expression there was in each tissue and time point. And so this is the total number of differentially expressed genes, which is a decent chunk of the genome. So things are not getting particularly easier there. But there's substantial divergence in gene expression, which is itself interesting in these metabolic tissues. But let me show you some of the things some of the clues we can get by analyzing these data. In the liver, Mark looked at 3 different time points, which means that he can assess the trajectory of expression, of differential expression. So for example, you think of these 3 different time points here. In this example, gene A has this kind of pattern this should be, so I have the, right, on the mainland along with gene B, so you can see maybe they're coordinately decreasing their expression throughout development. But then when you look at Gough Island mice, and this is data from a real example, they show a totally different pattern. And so it's these kinds of coordinated shifts in expression that get us excited. And so when Mark did that for the whole transcriptome, in each case in the liver, he found 66 what we're calling potentially co-regulated groups of differentially expressed genes. And there's kind of formal ways to assess this. that we're working with, but this is the basic idea. And just as a further example, if you pick out of those 66 one group, it has 38 genes in it. There are several genes, they show this pattern that I showed before, where you have this extension of the increase in expression over time in GOF, that despite a decrease in the mainland strain after 2 weeks. And that you can do a gene ontology analysis, which always tells you exactly what you hope it does. That was a little bit of a sarcastic comment. And you can discover, enrichment in this case for regulators of cellular differentiation, organ morphogenesis, and things like that. Anyway, the identities of these genes actually suggest to us a hypothesis that we're interested in testing now, which is that there's been continued differentiation of liver cell types in GOF mice after 2 weeks. Okay, so now we're doing more histology of the literature of the liver to try to take that apart and see if that's true. And this is a giant data set that we're trying to understand, but again, that's one of the clues that we can get. Now, one of the other things we can do is just ask how many differentially expressed genes in these key metabolic tissues and time points overlap with the QTL. And this is just a very rough summary of that. But the answer is a lot, because there are, I showed you, there are several thousand differentially expressed genes. We have about 13 to 14 total QTL. And so it's actually kind of a fun problem to think about how those should be overlaid under models that are less interesting. But we do identify genes that make sense in terms of what we know about them, that are located at the estimated position of the QTL, and so that gives us some candidates to work with. Okay, but in the last little bit here, I want to talk about, just mention population genetics. And we're still at the beginning of this project, despite having worked on it for about 8 years. But, as I said, something I'm learning is that whole complex trait thing, it's very well named. So, but I think it's exciting to try to take this apart. So we're also interested in the population genomics of island colonization in general. So what can we find the regions of the genome that contain the mutations that enable island colonizers to adapt to their environments? And we're working kind of in parallel to reconstruct the demographic history. And I'm not going to talk about this part, but I'll just tell you that after about a year of doing approximate Bayesian computation, we actually get estimates that make sense, but the confidence intervals are big, really big. And so I don't know, it's an interesting thing if the point estimates sort of correspond exactly with the historical record, but then you can't exclude things that happened a really long time ago or happened yesterday. It's, philosophically, there's an interesting question there. But we're working on some other, with some other methods that use things like identity by descent sharing to try to narrow that down. It's been harder than I thought it would be. But the other part of this is looking for evidence of adaptive evolution. So first I can just tell you, looking genome-wide, these are genome sequences from 14 GOF Island mice and 16 European mice. And here we're showing France and Germany together, but you get the same kinds of plots when you look at them separately. There's about a 20% reduction in the average number of pairwise differences, this measure of sequence diversity. This is actually looking at regions of the genome that we chose specifically for demographic history reconstruction, which are those in high recombination regions where there aren't a lot of genes around. So this is kind of our best guess about the neutral patterns. We also see a genomic skew towards intermediate allele frequencies. Both of these patterns are, of course, well-known signatures of population bottlenecks. So that gives us a sense that something's working. And the ultimate goal here is to develop a baseline model for demographic history that we can use to identify the genomic regions that are very unusual, because we're working with a population that's far away from mutation drift equilibrium. And so, you know, to just simply do look for outlying regions of the genome that show unusual behavior is not, I think, a totally fair approach. So having said that, of course, for this talk, I'm going to show you an example of what happens when you do that, because we don't have the demographic model yet. And this is just one example. We have started to scour the QTL regions for signatures of selection using a wide variety of approaches. And I'll just tell you that it's sort of mixed. You know, there are some regions where there are pretty clear signatures right on top of QTL, which also contain differentially expressed genes. And I think they're very good candidates. But then there are some QTL where we can't find anything, or the kind of signature I'm going to show you lands in a region that is far away from any genes or any conserved sequences that might indicate enhancers or whatever. The mouse genome is really big. But here's an example of the kind of pattern we see, which is, of course, totally representative, and I didn't choose it at all, just to show you one of the better examples. So this is a score on the y-axis that comes from an approach that compares the distribution of allele frequencies in a genomic region to what you would predict it should be by looking at the distribution of allele frequencies for the whole genome. And it's actually a modification of a test that was developed by Rasmus Nielsen, and it's quite powerful. So anyway, this is an example of the kind of thing we see. This piece right here, where this is one of the most extreme outliers in the genome here, and from some neutral simulations of our approximate demographic model, we don't ever get anything that approaches this, but we need to do more of that modeling. And so this is interesting regardless of what's located in that region, right? I mean, it could be involved in any form of adaptive evolution that may have to do with island colonization. But it gets more interesting when I tell you that this is on chromosome 13. You know, our QTL have broad confidence intervals, but there also are point estimates. That is the location on the chromosome where you see the strongest statistical evidence for the location of the QTL, and it's right here, okay? Now again, broad confidence limits, but it's here. And so something else we can do is just take a look at what genes are located in this region. There's a gene that is involved in a number of processes in the pancreas that seem relevant in terms of the regulation of insulin. And there's this other gene called PILO that basically this signature is just south of it, or distal end of the gene. And that gene, it turns out, I just learned this morning from my postdoc Mark, that it's one of the most highly differentially expressed genes in our comparison. And knockouts of this gene result in an arrest of growth, both embryonically and after birth. So what you may also know is that about half of the genes in the genome, when you knock them out, affect body size. So it's a little bit misleading. But what we hope is by intersecting these different lines of evidence, we can really start to whittle down what's happening. So that's what this slide is meant to indicate. We're looking for what's happening in here. And it's quite a complicated feat to go through all these data sets and whittle them down this way, but we are getting some candidates that I think we can pursue functionally now. And hopefully I'll have more to say about that later. But since this is the MVZ, I felt like I should at least try to address the question that's lurking behind this. So let's say we find all these genes and all these genetic changes, so what? Why do we want to know that in the first place? Well, part of it is that the genetics is part of the process that makes it possible to achieve body size. So in that sense, you're getting close to the mechanism. But I think another very interesting question is why? Why are mice getting big on islands? And I'm hoping that all of you can tell me the answer to that question, or some of you can. I already asked Jim Patton this morning. But just to begin to get a little perspective on this, my former student, Lauren Brooks, went through the literature and found all the islands where this subspecies of house mice we're working with, Mus musculus domesticus, have recorded body weights with a minimum sample size and some standard deviation to look at. And then she went through these online databases looking at environmental gradients, just about anything we could think of. And this is just for island populations, okay? A lot of you all know, and some of you have done this kind of work, that it's been done a lot going across species. And what we're interested in doing here is all within a single subspecies. So we're hopeful that phylogenetic correlations here are not going to confuse the sorts of patterns that we find. Anyway, when she did that, she found a number of associations. Many of them were weak. We're looking at about 40 islands here. But the biggest one by far was this. When she compared islands that had predators, either avian or mammalian predators, to those that didn't, the mice on islands that didn't were quite a bit heavier. Okay, Gough Island mice have no predators on the island. This doesn't, of course, prove anything about what happened on Gough Island, but I think it's an interesting lead. And this kind of a pattern has been seen in the ecological literature before, but not as much looking within a species. Okay, so that we're interested in pursuing that too. So just to wrap things up here, I showed you in this case of the island rule that size evolution targeted early acceleration of growth, and it is indeed heritable. We can locate the genomic regions that were involved, and size evolution involved multiple mutations with modest effects. They're mostly additive. Skeletal evolution used mutations with a mixture of global and local effects. We found substantial evolution of gene expression, but also signs that it's quite modular, which points us towards some of the underlying processes. Population genetics works. Signs of island colonization are visible in genomic variation, although I'll admit here that our working model is that most of this adaptation we're looking at, if it's adaptation, was actually coming from standing variation. And if that's the case, and we're looking at a trait that it has some polygenicity to it, it may be that looking for selective sweeps is not going to be very helpful. And so that's something else we're wrestling with, is this stratum between population and quantitative genetics, and how to combine these different pieces of evidence. And at least in this comparative survey, the loss of predators in particular was associated with increased size evolution in island mice. And lastly, I'll just mention that I do understand looking at a rule through the lens of a single example is maybe limited. And so the point of working on house mice is that now that we've identified QTL and are drilling down on the genes for Gough Island mice, we can move on to other islands with the hope that that will give us even more resolution and open up the possibility to understand the island rule itself. So if you have ideas about which island we should move towards next, we're definitely taking nominations. Thank you very much.
All right, if somebody could hit the lights, got time for questions. We've got in the back there.
I see you.
I'm wondering what proportion of the changes in gene expression are due to changes in cis versus trans?
We have not done the cis-trans kind of test, and that's something we're interested in doing. It would be really interesting.
Have you identified any trade-offs for the big body size? And did you look if any of the QTLs that were associated with large body size was also associated with whatever trade-off if there was any?
Wait, so I didn't get the last part.
If it was also associated with the trade-off, if you
Yeah,
identified
we haven't done that. We're just now moving into characterizing life history traits, behavior, and so there may very well be trade-offs, and we're actually talking about doing more crosses to identify genetic correlations between these different traits, but we haven't done that yet. I think it would be quite useful, especially to figure out if there's a selective regime here, what is it? Yeah.
Is it possible you could find an island where you could reintroduce cats and follow the consequences?
That would be great. I don't know how feasible that is.
Yeah.
But it would be
Maybe
great
on a
to see.
small enough island somewhere in the middle of nowhere. You see all the islands off the west coast of Australia. It
Yeah.
struck me that that might be a good place to look.
Yeah, that's a good idea.
Well, the reverse of that question is there's a lot of effort on island conservation to get rid of the cats. And so what happens to the mice after that happens? I mean,
Exactly.
There's a big program at UC Santa Cruz and elsewhere to do that in the Gulf of Mexico and Galapagos and the Channel Islands and so forth.
Yeah. No, that's a good idea. Eileen?
Brett, you may not know the answer to this yet. Do all mice get big in the same way? Are there,
You mean mice on different islands, or
Yeah.
mice? We don't know that.
okay,
There haven't been.
just a comment. The other thing.
Yeah, there haven't. Oh, go ahead. Let me.
Well, no, I'm gonna,
Okay.
you finish it and I'll.
There haven't been a lot of these common garden experiments where you get, and as you probably know, body size has a significant environmental component. And so our QTL here are explaining actually most of the estimated genetic variance in the trait, but you may have noticed the mice got smaller in the lab. A little bit smaller. So yeah, we don't know that, but that's something that I've been thinking about is even if we can't do the genetic mapping on other islands, it would actually maybe be more informative to just take, following the Nachman approach, to just take mice from a lot of different islands and watch them grow in a common environment and to answer that very question.
Jim, you wanted to weigh
Well, I was gonna ask something related, but I think somewhat different question, and that is, do you have any idea at all as to where those mice on Garth came from?
Yeah. Well, the idea,
So, I mean, the sealers were all out of Great Britain, for example, or?
well, so the history, the shipping history, and our microsatellite work that we did a few years ago suggests that they well, it's pretty strong evidence they came from Western Europe, but it's actually, with microsatellites, we found it very challenging to differentiate between different countries. And also, there's an interesting question about whether it was a single colonization or multiple colonization events despite being a pretty remote island. And so right now we're looking at dense genotypes from across the genome, and I think we may be able to answer that question. But it's really important to know that because we don't even know, I mean, your whole story about what happened here really changes if, for example, the mice that colonized Gough Island were bigger than average to start with. Or if they were
Well, that's what
smaller
I was
than
gonna
average.
ask,
Yeah,
what do you know about, kind of standing variation in body size or anything else from the potential source areas?
Yeah, you should ask
Yeah.
him. He's been measuring that,
Yeah.
at least in North America and South America.
You got some question in the back?
Yeah.
What percentage of the body size increase do you think is due to behavioral changes versus developmental changes? Like, do the mice from Gough Island just eat a ton of food? Do they work out? Do they sit 9 hours in a cage? Like, just like by looking at them, do they have any obvious behavioral differences, or is it all developmental?
Yeah, that's another great question. So remember that these differences in size that we documented are in a common environment. And so they're eating the highly natural diet of Purina mouse chow. Everybody's like, don't do that. But that's, it's a standard diet. And so the differences in growth persist on a common diet.
But they might eat more or less.
That is actually what we're starting to test now. So we just purchased these fancy metabolic cages in which we can monitor how much food they're eating, how much water they're eating, respiration rates, all of that kind of stuff. And so we're interested in looking into that. But we don't know what role, if any, for example, the apparent shift in diet has had. That people, the avian biologists who are actually working on the island and catching these mice, they think that the diet is a big deal. And there is some sort of anecdotal evidence that during the seasons when they're eating the most meat, they're bigger. So they think that that's really important. But we're actually about to go in that direction. We're being kind of dragged into studying behavior by its importance. Right? And so we had a student that did an open field test, which doesn't tell you much about diet, but and discovered that the mice from Gough Island show a much greater propensity to explore than mice from, at least the reference strains from the mainland. And so that is something we're interested in following up on and maybe even trying to understand the genetics of that. Oh, go ahead.
I had a kind of a similar question about seasonality in nesting behavior of the birds and how that might influence fluctuating or introduce fluctuating selection.
Yeah.
On the sort of, you alluded to that. Hard to identify the genomic signatures of that, of course.
Yeah,
but
that's right.
and then I was going to ask is that, I remember being in Galapagos and seeing brazen rats and mice during the day. And so I'm curious if their diurnal versus nocturnal behavior is shifted.
There isn't a lot of evidence for that. This hunting, is happening at night. They seem to be mostly nocturnal. But I think that's an interesting question. And I don't know how much how high the resolution is on that, how much they've looked at the activity patterns as opposed to just trapping mice. There are some, if you're feeling brazen, there are some interesting videos online. If you just look up giant carnivorous mouse. But those are all night cameras. So I don't know, but that's a good question.
Brett, I have a question. So the QTL sign test suggests that there's positive selection.
Yes.
I mean, I understand it's marginally significant. And then you're looking for selective sweeps, so you're looking for signals of positive selection. But the correlation with predators sounds like a release of selective constraint. So when cats or other predators are not around, all of a sudden something that was formerly bad is now okay. It's okay to grow bigger. And so you might not expect to see a signature of selection.
Yep.
What do you think the selective agent is?
I don't know. That's something that we spend a lot of time arguing about in the lab because you're right. I mean, it could be that there's normally stabilizing selection on body size, and then you basically remove one of the agents of that stabilizing selection from the upper tail, and they just wander. And I don't know if that would leave a signature in the genome. Probably not, but it depends. I mean, then there's this. We don't have a way that I know of to differentiate those two. But it could explain, if we don't find a lot of selective sweeps in QTLs, that could be a reason why.
I like Dale's idea of introducing cats.
Yeah.
If that were the case, if it was just like relaxed selection on these things, what would you expect would happen with just lab mice that don't have predators? Would they change, or would they not be? Are they not subject to this balancing selection? Or?
To the stabilizing selection?
Yeah.
So laboratory mice, you mean like the colony that we set up, or do you
Sure,
mean
just
like,
any. I mean, if it turns out that body size is being constrained by predators in a field setting, and then you remove one of the predators, a top end predator, and they get bigger, would you expect the same thing to happen in a lab setting when they don't have predators?
I think you would. I think the prediction in terms of reducing stabilizing selection is that you'd see an increase in the variance in the trait, and whether that's directional or not depends on how close the mean of the distribution is to the lower bound. Does that make sense? So I don't know how to articulate that better, so maybe I won't try. But I think you should predict that laboratory populations without predators should have an increased variance over time. But I'm not sure how quickly that would move you in the direction towards getting bigger, right? I mean, yeah.
How fast did it happen in this system? Did people document giant mice like right off the bat once they were?
The
aware of
first
them?
records are in sort of the middle of the 20th century, and so that's probably 150 years in. So we don't know how fast, but we do know that there's evidence that people have weighed them in multiple time points, and they're still getting bigger. So evolution is ongoing.
Maybe one last question here.
Well, I was wondering, have you tried to study what the ecological variables are that favors a larger size? In other words, you just don't get bigger because you couldn't, there probably is also some selective advantage in terms of obtaining food or temperature control or, what,
Yeah. I'm afraid the only attempt we made was that kind of comparative study. So we really haven't done anything more experimental. Is that kind of what you're? But there.
Well, again, if you're going to try to duplicate this in the lab,
Yeah.
you have to change. You have to also make sure that the things that favored a larger size are present in the lab.
That's right.
Or they're not going to be getting larger, even though there is no cat there.
Yeah. No, I think we're kind of heading in that direction. There's been a little bit of challenge with animal care, and not everywhere lets you have enclosures and things like that. And so but it would be great to do that. It would be great to do some experiments and introduce and remove variables and see what happens. That would be great.
All right, let's thank Bret once again.